All right, so we are looking at the gene for, or the map for a fluorescent protein called mCherry. Now what you'll often see with genes, or what are called cDNAs, is the actual gene that gets expressed will be here, okay? So meaning this part will be which codon? What codon would we see there? Remember codons from a few weeks ago? Fuzzy? Not so much? Okay, we'll probably review that. A, U, and G. Okay. And at the end here, there's going to be the stop codon. And everything in between here are the sequences, the AUGs and Cs, that will tell us to build that mCherry protein, right? But what you notice is that there's this region upstream, we call it, and a region downstream, and that varies from gene to gene, past that arrow, that's a little bit extra. Yes? So far so good? So you'll always have, like, think of like, you'll have a bit of a lead fuse, and then you'll have a little bit of a fuse after the stop codon. For every gene that when I give you your gene, you'll see that pattern, okay? So the AUG, or the start codon, is not necessarily the beginning of the DNA fragment that you get. And that's a good thing, because that allows us to do some splicing without chopping up our gene. So our task is to take this mCherry gene and to put it into one of our vectors, which means we need to pick an enzyme from this first section, our first cut. Now, ideally you want to pick an enzyme as close to that start codon as possible, because you want your fragments to be as small as possible when you splice them into the plasmid, because plasmids, when they get to around 15 kb, they start to 12 to 15 kb, bacteria don't like them. Okay, so plasmids that are synthesized don't go into bacteria so well, and you run into problems. So again, it's basically being as conservative and tight to the wire as you possibly can be. Right? Which enzyme did we pick for that? Some of you picked, most of you actually picked the correct one. It was awesome. I was so happy that you got this. Shout it out. Bam! Oh, you went to XBA? XBA is at 80. BAMH1's at 86. XBA is not wrong. It could work. You're only 6 base pairs. But if we're really going super tight, go to BAMH1. But that's fine. And as long as you got it to work, then it worked. Okay? And it would work if you actually did it in the lab. So we'll call that restriction endonuclease number 1. Okay, so that's our upstream. And so it's BAMH1. And it's at position 86. Good. Cool. So at that location is a cut site. Now, what's really cool is from this point forward, we don't need to know what the restriction sites are, because now you know from the previous activity that they're always going to be the same no matter what, right? And if we need to look them up, you can look them up. You don't need to memorize those. It's kind of like memorizing the periodic table. It's a silly thing. Knowing how to read it is more important. Okay? So I know if I cut this thing with BAMH1 and I cut the plasma with BAMH1, they're going to match up. That's the whole point to this. That's how this works. All right? What was our downstream enzyme? Yep. Sal. Little buddy Sal. Okay, where's Sal? Where's Sal? There it is, at 812. So we'll call that restriction endonuclease number 2, Sal 1, at 812. Okay, so just for perspective then, the fragment we're going to keep, I'll take my crayon out because that's really high tech. Is from here to cell 1, which is there. BAMH1 to cell 1 there. Okay? BS. No comment. Okay. No crap. All right, so we're going to cut that fragment out. Just got it. Just got it. Now, there's another enzyme I'm going to add. So take this out from yesterday. Take your map out from yesterday. We're going to level things up. What we want to do is choose an enzyme that cuts inside of that, but also inside of pBluescript. Okay, and I'm going to give you that enzyme right now. pBSSKmCherry. We're going to call that FP for fingerprinting. So what's that 331? You're free to take out your calculators to do some numbers, okay? And if you want to use your calculator on your phone, that's fine. So I'm going to be consciously aware that BSA cuts closer to BAMH1 or to the beginning of my gene than it does to cell 1 over here. Okay? We're not going to keep these end fragments. That's why we're cutting it. And this is just like the literal cut and paste with the green strips of paper and the blue strips of paper we did. Same idea. Exact same idea. Okay? Except the difference is this is a real gene. That other one I just made up. Okay? So, how big is our fragment totally? Well, we're going to go 812 minus 86. Gives us a fragment, or an mCherry fragment, now, when you're doing genetic engineering, you call that fragment that you're going to put into a plasmid an insert. Literally, it's called an insert because you're inserting it. All right? So that thing is 726 base pairs total. From BAMH1 to BSA-A1, so we're going to go 331 minus 86 is a fragment that's 245. Whoa. Let's choose a better color. Black. So from here to here is 245. Okay? From cell 1, 812 to BSA-A1, 812 minus 331 is a 481 piece. Everyone cool so far? So cell 1's is 812. BSA-A1 is 331. So I just want 812 minus 331 is 481 from here to here. What was your question? Sorry? Yeah, like you could have got that if you went 726 minus 245. That would work too. Right? It has to. And that's a double check you can do. I recommend it. In fact. Okay. All right. So our fragment then in our mind's eye is a fragment that goes B to S from start to finish. And yeah, we got a little B in there. Okay? So I just want to keep that in the back of my head when I'm doing my inserting. Now I got to pick my vector. My vector is another name for a plasmid. Okay, so if you ever hear that, they'll call it a plasmid vector, or sometimes they just call it a vector. The vector meaning it's going from here to there. That's what the definition of vector is. It's something with magnitude and direction. Meaning, we've got this plasmid that's going from here into bacteria. All right? So that we're using the plasmid to carry our gene into bacteria. Now we can make this protein that glows, and maybe we can, I don't know, attach it to something medically relevant, like labeling tumors or something useful like that. All right? And what's really cool about these biotech tools is now that you're aware of them, when you start learning about this or you start learning about a gene, ideas just come from people going, hey, what if this? And hey, what if that? What if I took this cherry thing and I attached it to that? Now it's labeled red. Now wherever that protein is made in the animal or the organism, it'll glow red. And now I know what tissue that protein belongs to. Because we don't always know, right? And that helps us to understand what proteins do. So we have two choices, KS or SK. How do we choose? Well, remember, we're using enzymes that are going to be spliced within this MCS. What does MCS stand for again? That's right, multiple cloning site. And what's really neat about the MCS is it's actually built into this other gene called LAC-Z. And what does LAC-Z gene tell us or give to the bacteria? Blue staining, blue-white screening, right? But if I shove another big gene in there, can I still stain blue or no? So we're actually looking for a loss of that trait. So if my bacteria are glowing blue, or they're staining blue, they don't glow, but they stain blue, right? Then I know that it's just pBluescript plasmid that went in. But if they don't stain blue, and they're still there, I know that they have made it through, and they probably carry my insert. They probably do, but I don't know for sure. They might, okay? So I would grow a plate of bacteria, a bunch of them would stain blue, and a bunch won't. Bless you. Okay? Now, inside of that plate, that little growth plate, like our friendly butcher guy was growing in that little video, is another substance. What is that substance? I'll give you a hint. It's a substance that kills bacteria. That rhymes with penicillin.
Penicillin.
Not penicillin. Nope. Ampicillin. Okay? So we're growing our bacteria, and in the bacterial dish is this antibiotic called ampicillin. Now, without ampicillin, any bacteria will grow, which means that whole dish would be covered. And you'd see blue splotches, and you would see, it would be blue, a mix of blue and yellow. They're kind of an off-white, sort of a pussy color. That's what the colonies grow, all right? Sort of gross almost. Not that gross. I mean, there's grosser things. But without the ampicillin, this plate would be totally covered in what they call a lawn. But since we put this ampicillin antibiotic in there, only bacteria that what? Are resistant to ampicillin. Okay? Now, under normal conditions, they're not. But these ones that have grown, I'm sure that they have what inside of them now? The gene. Okay? And that gene is over here. So far, so good? Now, there's this cool thing up here called the promoter. And that's just basically like a switch that tells the bacteria, hey, make this ampicillin-resistant beta-lactamase enzyme. Okay? So, for example, you were looking yesterday at using BPM1, right? You were looking at using BPM1, which theoretically you could splice, but then you would lose that antibiotic resistance, which means you wouldn't be able to pick an individual bacteria colony out of that dish. Okay? So that's important. We always use the multiple cloning site because then the only thing we're going to lose is that blue stain, and we want to lose that blue stain. If it's blue staining, then we know it's just plasmid in there. If it's growing, but it's not blue staining, then we know it's got the ampicillin resistance and it probably has our insert. And again, I'm going to use that word probably. It's important because we have to verify that later. Okay. So what we do is we got to figure out now which version of pBluescript we should use, KS or SK. Remember, our insert is going from B to S. All right. So if I look at KS, and I look for cell 1, there's BAMH1 there, so there's my B, and then there's cell 1 there. Which means that if I inserted this, which direction is my gene, my mCherry? going? Clockwise or counterclockwise? It would be pointing clockwise. What would be the problem with this? It's going backwards. How do you know that? The arrow's going backwards. All right? So here's your LAC-Z gene. So what we're going to do is we're going to shove mCherry gene into that LAC-Z space that's going to kill LAC-Z. Now, if you look over here, you've got this thing called the LAC operator and the LAC promoter. Okay? That area up there, those little components are actually the control switches for everything that's downstream, like what's next, the next gene. Okay? So if I put that in, it would make that protein, but it'd be the reverse. I'd be actually making the antisense backwards, which would not get me a glowy protein. It would get me something else entirely. It'd be useless to me. Okay? So are we using KS? Ew, KS is out. All right, so we want our mCherry arrow to go. Now, what you want to do is you want to make sure you're going to verify. Okay? And this is where I struggled at times because I would cut corners here and there. Because I was a little bit lazy, I'll be honest. Just like a student, typical student. You know, don't want to put in that little bit extra effort. But it catches up with you later. Okay? So, let's verify up. Cell 1 is right here. At position 674. And BAMH1 is further down, right over here. At position 719. And if I jam my mCherry gene in there, insert, am I going in the right direction? Yes. Does every plasmid go counterclockwise? No, not necessarily. So you have to watch that when you're building and designing these things. All right? Really important to understand the tools that you're using and the way they need to be used when you're doing this. And you might be doing this in the future. It's very possible. This technology is kind of like workhorse level technology now. It seems very new and it's not. Okay? It's decades old, actually. But it's kind of the basics. All right? We got to keep the ORI intact. That's got to be intact because that is giving the bacteria instructions to make a bunch of copies of the plasmid inside the bacteria. Because eventually what we do is we extract this thing from the bacteria. Or we get the bacteria to make protein for us. Okay. What's next? Sizing. I've got to figure this out. So I've got this position of cell 1, and I'm just going to kind of extrapolate it over here. All the way around to BAMH1 over here. And we can just kind of scribble that in. Happy little fragments. Where's BSA A1? Up top, right? Right there. At which position? 237. Beautiful. Okay. So that means for later, how big is this piece here? How big is that piece there? Okay. How'd you get that? 674 to, and that gives us? Okay, so from here to here is 437. And the little chunk that we're going to take out, 719 minus 64. How big is the chunk we're gonna remove? from here to here. How big is that piece? That little chunk we're going to take out. So 719 minus 674 is 45. So 45 out, and we're going to pop in that other piece. All right. So that's one chunk there from BS to BSA 1 to Sal 1 is 437. Now we got a whole other chunk going all the way back around. And don't worry, I'll color it all in. From here to here. How do we figure that out? For this big dude, I'm just going to go 2958 minus the 45 we're going to throw out minus the 437. Can someone run that for me? What size is that? Sorry? Oh, because I'm just measuring the distance. So we're going from Sal 1 to BSA A1. So then I want to know how big is the piece that's going from BSA 1 all the way back around to BAM H1. You see that now? Okay. This looks horrible, but that's okay. 2,406? 2476. Okay. Cool. Now what? Yep.
Sorry, we're going to cut the mCherry.
Sal 1 and BAM H1. Right. So now let's get our fragments put together. Let's put this guy together. All right. Let me get my pieces. Okay. So there's my insert map. There's my gene map, or my plasmid map, I should say. We can kind of redo this a bit. Oops. So it's a bit cleaner. Sal 1 is at 674. BSA A1 is at 237. That is 437 there. This is 2,476 here. MH1 at 719. There. I'm going to kind of color in the fragments we're keeping. Okay, it's a bit cleaner now. All right, mCherry fragment. I'm going to cheat and do a shortcut map. So if I go back to our original, 245 and 481. So I'm only looking at the keeper fragments here. Who can tell me what site's here? What sticky end's here? On this end. What sticky end is that? If I've cut my mCherry original DNA out and I've got the fragment that I'm keeping. Yeah? Yeah, that's the 5' end. But what sticky, like what enzyme is there? Yep, thank you. Okay, and who's going to be having a cut site right there? BSA A1. And on this end, cell 1. So I'm just going to put that information over here. Give himself another plasmid. So, what's my total size then? Two, 4, 7, 6, 4, 3, 7, 481, 245. Let's add those up. Three, 6, 3, 9. Yeah. Can I get your attention, please? Thank you. Okay, next step. We need percentages for each of these fragments. So 2476 divided by 3639 is 68%. Almost 70. 437 divided by 3639. 12%. $41 divided by 3639. 13%. And it may not add up to 100, but that's fine. This is just a ballparker. Okay. 245. Divided by 3639, 7%. Now we got to position things. So BSA A1, that's going to be our first site, right? So its number doesn't change. So it's still at the blue BSA A1. It's still at 237. So 237 divided by 3639 equals 7%. So how do we tackle that guy? Well, I just go there and there. That's 25%. That's 12%. 12.5. So that's 6.5, 6-ish. So about there. Still at 237. Cell 1, 674 divided by 3639 is 19%. So now I'm going to take BSA-A1 from here and go 19%. So we go across to 50, 25, come back to about there. No, that doesn't make sense. No, 18 of the total, sorry. There's 25, so bring it back up here. So in here's our 437 fragment. Who comes next? Yep, the mCherry. Which part? 481 or 245? 245, so we're going to go from BAM to BSA. No, we're going from cell. So this cell is going to connect up with that cell. This BAM is going to connect up with that BAM. So from this cell at 674, what's the next fragment? 41 or 245? 41. Okay, so we're going to go 481 divided by 3639 is 13%. Okay, so we want to go about half, half, half. So about there. What enzyme name goes there? Yep. And now we're going to our next fragment, which is BAMH1. Okay? So our next fragment is the 245 fragment. And we're just going to color them. So that's 7%, right? From before, there. About there. And what we can do even is go over them in purple, where they join up. Cell 1. What's missing? Something's not quite finished. Okay. What's missing? Yes. Yeah, for who? Okay, the total. So we want to throw the total in here. 3639. Whoop. We also need the name pBSSKmCherry. You could even call it MC for short. That'd be cool. No problem there. What else is missing? One more thing. Well, two more. Two more little bits. Cut sites, yeah. So BSA is now at, yeah, 1155. And I got that by just adding 41 to 674, right? Plus another 245, 1400. So there's our recombinant map. Now, what we could do is do a bunch of digestion. So if we treated this thing with BSA A1, how many times would it cut? How many times would this new combination be cut if we treated it with BSA A1? Twice, right? BSA A1 enzyme would cut here and would cut here. And that would generate this weird combination fragment there and another combination fragment there. That never happened before, that never existed before, wouldn't it? You look lost. Did I lose you? A little bit? Okay. Go with me on this. Remember, the BSA A1 is a location site for an enzyme to cut. Okay? So I'm going to draw this guy. And I don't know if he's a 5' cutter or a 3' cutter, but he's a biter. Okay? So he's going to go along and scan along this DNA, right? Where will he bite? Give me my two locations where he's going to bite. It's going to recognize here, and it's going to bite right there. So if this guy here, bites there, and then scans along, looking for the same site, the same sequence, it's going to bite there now, isn't it? As well. Twice, right? So if it bites here and it bites there, now I've got a fragment that's going from here to here. Do you see that now? Okay, so that's going to be how big? 900 and something, right? So 437 plus 41. Has that combination of 437 and 40, have they ever been together before in the history of the whole wide world? No. That's how we prove that it worked. Because I'm going to generate that piece, which means that's the small fragment. Okay, we'll call it fragment 1. And that's going to, I'll just use a different color so you can see it better. Oh, yellow, I guess. And that's going to leave this other big piece from here to here. You see that now? Is it clear? Cool. All right. So that's why we have a fingerprint enzyme. Because the 245 should be stuck to the 2476. Had things gone backwards or we used the wrong plasmid, it would have gone the other way around, wouldn't it? And it would have come out differently. So you want to always verify that what you thought you made, you actually did make. All right? You could go so far as to actually get this join-up section sequenced and prove that the DNA in there actually has that connection, like reading the letters with the sequencing that we did. Okay? But that'd be expensive. It would be very expensive. And we generally don't do that. Okay? So that's how to make a map and how to read a map, build a map, and then use a map. And now we have a plasmid that will make our bacteria do what? Have which two superpowers does it have now? Yep. Glow red and, ow, that had to hurt. Ampicillin resistance. Okay? Pretty cool. we could use those bacteria to then make this mCherry, and then we have it for whatever purpose we want. Okay? The other alternative then, once we have this, is I could then splice another gene right in here, so the front part of that protein would have a little tag that glows, and then anything downstream from it would be your original protein. Okay? So that's how they actually do that. For real.